And we move on to our next speaker. So this is Pauln Martin from the University of Bristol. And as I said, Paul goes back a very long way with David to the ICRF in Oxford. And the title of his talk is Mostly Down in Cornwall.
Yeah, so most speakers today have been gifted a sort of era of David's career to talk about. And I'm gifted with talking about his holidays in Cornwall. But Ros insisted on me topping and tailing the story with a bit of science, and so I'll do that. So I first met David there when Julian Lewis there, who was my PhD supervisor, dragged us from, this is Gavin Swanson and myself, who were the two folk in Julian's lab, dragged us from King's College in the Strand midway through my PhD to this amazing new institute, the ICRF Developmental Biology Unit in the zoology building in Oxford. And it was a hugely exciting place to be. You know, all the top PIs from all the top model organisms in developmental biology, and we brought kick-ass chick, Julian and my lab, and there was no need to laugh then, actually. So Julian brought a chick into the game. And I was just sort of a lowly PhD student, interacted not a great deal with David, although inspired by his talks, and I interacted quite a lot with the folk in his lab. But towards the end of my PhD, Julian and I published a paper. This is in the days when you could just publish an observation in Nature. And we showed that if you wounded the chick embryo, Julian worked on chick limb development and ear development, and we'd do cut and paste surgery. And what we noticed was that if you make a wound on the dorsal surface of chick embryo's limb bud, it closes, the epithelium closes with a little contractile actinomyosin purse string that you can see here. This is one of the sort of early confocals, and we showed it by EM as well. And this thing contracts and draws the epithelial hole closed. And I remember David saying, this is reminiscent of dorsal closure in flies. Dan Keyhart, I'd gone to this is David saying, I went to a talk by Dan Keyhart a little while ago, and he sees exactly the same, Paul, at the tail end of fly embryogenesis as a fly is about to transition into becoming a maggot. You should read Dan Keyhart's papers. I did. And when I set up lab, for quite a while, what we did was live image dorsal closure as a model of wound healing, because in chicks you can't do dynamic studies, there's not very good genetics. So David sort of set me up with one of the areas of research that we ended up doing, and we work now in mouse and fish, but also in flies. And I'll finish the talk off, I promise, Ros, with a tiny bit of science. Quickly tell you, when I left the lab, Julian Lewis, my PhD supervisor, and David had a blossoming collaboration. And that's what the unit was supposed to be about. You bring all these model organisms together and they marry each other and interact with each other. And it worked a treat for this is Julian and David. Initially, I couldn't find any photos of them together. Actually, I moaned about this a week ago to Phil, and he found me a slide. I'll show you it in a second. But essentially, this epitomizes their relationship. They had very complementary skills. Julian was quite a shy man, a vertebrate neurobiologist, developmental neurobiologist, and an amazing mathematical modeler. Less shy, David Ish, a Drosophilist, very keen on technologies, as we've heard, and also fantastic at networking. You marry those things together, which this slide that Phil gave me does. Can you see? Their jumpers emerge, their beards emerge, their glasses. And you'll notice at the back there, another little head trying to merge in. That's Domingos Enrique's head. And he was the glue that sort of, got these two labs working together. And essentially, what they did was flies, Notch delta signaling, neurogenesis. They learnt from that. Some vertebrate orthologs of those genes had been cloned. Not much was known about what they did. And they figured out that in chick, they were involved in neurogenesis. Domingos cloned several genes and did in situs. Can you see pepper and salt expression in the neural plate, bit of loud expression in the pre-Semitic mesoderm as well. I said David networked a lot, so they did this in chick, but they collaborated with Chris Kintner's lab and did it in Xenopus too. And until Julian died about 10 years ago, and in the year he died, they were still publishing papers. So this wasn't a neural plate stuff, but this was oscillating cells in the Semitic clock. So their collaboration was fruitful and impressive, and happened after I'd left the lab. I'm going to talk about holidaying, because that's really where I got to spend most of my scientific interaction and personal interaction time with David. So Kate knows, this is her, she's my wife. For about 25 years, we've been holidaying in this place, this same cottage in Cornwall there. This is a picture by our mutual friend, Jonathan Clark. And this is Kate. So about 20 years ago, David and Ros started coming to visit for a few days each summer and lived with us in this cottage. They liked it so much that they began to rent this tiny weeny, I mean, it's a shed almost, and they've been staying there for 20 years. This is its floor plan. There's nothing to it. There's a bed, a bathroom and a little table, but what it has got is windows. And that glass looks out, this is there, it's called the workshop, looks out on that. The bed is a favourite place for our dog Molly and my grandson Douglas. They don't have a garden, so they share ours. Here's the view. Here's our beach. This is essentially David's and my private beach. You see how idyllic it is, loads of lovely coves. So David, initially, 20 years ago, was not a good swimmer, a breaststroker. Ros also was a breaststroker, but they taught themselves, or maybe someone taught them, front crawl, and became quite competent, good swimmers. And I loved swimming with David off this beach. We didn't only swim. I'm mad keen on fishing, and I would take these guys fishing. For 2 or 3 weeks every summer, our 2 daughters had second parents, David and Ros. My youngest daughter, Charlotte, even used David as a sort of fashion role model. Maybe not the wisest choice of fashion role model. Here is David catching a fish. This was a small conger eel. He never caught. Our goldfish is bass, sea bass. They're very tasty. He never caught a bass, but Ros is much better angler than him, caught several bass on trips with me, and these are bass that she has cooked, which brings me on to something that we do more of than swimming and fishing, and that is cooking and eating. So this is Ros guiding my grandson Douglas how to bake biscuits. He loves Ros's biscuits. He was supposed to be learning how to bake those biscuits, except you'll notice his attention has been caught by David. And David knows a lot about everything, and if he doesn't know something, he'll find it out. Douglas had just asked him about sourcing a very special Lego thing, and David was finding out where to get it. Okay, distracted completely. I said that David and Ros didn't have a garden. They haven't got a garden in their little shed, so they share ours. And we, most lunchtimes and evenings, this is the last time David came to Prushkov. We also like potting. So these guys are the potters at the end of our road, at the end of the lane that leads to our cottage. This is Peter Swanson and his wife Annie, and they're leech inspired. You can see David and I outside waiting while Ros chooses a pot. Sometimes we had to wait quite a while. Right, so we did lots of chatting, David and I, often on holiday. This is a few weeks before he passed away. This is their, you'll know their house is up here, and we're walking down, discussing stuff, back to science, because he'd asked me, what our latest stuff was. And if I had fishy stuff, I would tell him, but I had some fly stuff to tell him about. It was a paper that we were just about to submit, and I need to set the scene a bit. Fly pupae, once you've taken them out of their pupal case, are translucent, and you can make lovely movies. I'm going to show you a wound made with a laser beam, and these cells here, these green ones with purple nuclei, are fly inflammatory cells. Okay, they're hemocytes, and this is what they do when you make a wound. They're rapidly drawn towards that wound. And we, for several years, my lab's been interested in the damage signals that recruit immune cells to wounds and how the immune cells respond. But we're also curious what they do, because in all model organisms, we care about what the inflammatory response is doing beyond just killing off bugs and clearing away cell and matrix debris. And one tissue we were particularly interested in, what the inflammatory response might be doing, is the wound epithelium. So this is the wound epithelium here. That's the front edge, and it extends back. And Yan Lan Mao's lab and mine have been very interested in what happens in those front few rows of cells. Nice actin cable assembles, phylopodia. There's a bit of shuffling amongst cells back from the leading edge. But back from that, there's some cell division to compensate for all those cells that were lost by the wound. But they're hard to spot. And so Jake Turley in my lab, who was a PhD student, trained initially as a mathematician, developed some deep learning tools to hunt down dividing cells. And we could use those two tools then to find these dividing cells, which you can see here. They find them, locate when and where they're happening and their orientation. And once you're able to do that with deep learning, you can crunch through many, many movies, many more than biology or an eye alone could do. And this is what he saw. So the cell division doesn't happen in the front few rows of cells. It happens back from the leading edge. It happens about 50 microns back from the leading edge. And you see a proliferative surge about 100 minutes, 2 hours post-wounding. Once you've characterized a wild-type wound situation, you can look in mutants, because flies offer fantastic genetics, as you've heard, and you can knock out various potential signals. And one Jake knocked out was those inflammatory cells. So are those inflammatory cells involved in driving this proliferative surge? He knocked these guys out and hugely dampened that proliferative pulse that happens 100 microns, I can't remember now, 50 or 60 microns back from the leading edge. Not completely. There are other signals. There's a massive calcium wave that spreads back from the wound leading edge. But a big component of the signals that drive proliferation in a wound edge epithelium comes from these guys. Okay. So I told David this, and he didn't tell me what to do next. So I've got to figure it out for myself. I'm going to miss his scientific insights lots. I'm going to miss him more down in Prussia Cove.
Thanks, Paul, very much. Anyone have any questions?
About swimming attire or Lego?
Question.
So this is probably a very ill-informed question because it's not my area. So is the mechanism you're describing part of why how the neural tube closes in the fetus?
Yeah, so it turns out, in fact, that wound healing recapitulates morphogenesis, and that closure of the neural tube involves a slightly differently, oriented bunch of actin purse strings. But when they meet, they knit together with phylopodia, just like in wound closure. Yeah. In fact, yeah. So during embryonic development, everywhere where tissue fuses, the same machinery is used as closes a wound.
Thank you, Paul. Thanks again.